All right, guys, this is going to be a long one, but I am actually condensing this from what it was before, so you're welcome. All right, so we've been talking about ATP, and we got into a little bit of cellular respiration, but this is going to be cellular respiration in detail. And my recommendation to you right now is to take a deep breath and not feel overwhelmed about everything that you're about to learn, because I promise you, we will break this down in every way possible in class. Okay. And before you know it, this is going to be your easiest and most favorite topic. I really promise you that. Okay. So, harvesting chemical energy. And we can see a bunch of pictures up here. We have our ATP, we have our mitochondria. This is what ATP actually looks like. Okay. This is an actual picture of the mitochondria, what it looks like. So, let us begin. Just kidding. It doesn't want to move forward. Okay. So what's the point? The point of cellular respiration is to make ATP. As we learned previously, ATP is our energy currency. It gives us energy when we need it. It does not store it long term. ATP is needed for every single process in life. It's how your brain functions. Okay. It requires a significant amount of ATP. So let's get going. All right. So energy is stored in our organic molecules. So what we've already learned about are carbohydrates, our fats, or proteins. Heterotrophs are organisms that have to eat those organic molecules or food. They digest organic molecules to get raw materials for synthesis. Okay, fuels for energy. There's a controlled release of energy, and the burning of fuels happens in a series of step-by-step enzyme-controlled reactions. Okay. So heterotrophs are what we are. Everything that eats is a heterotroph. We will be talking about autotrophs next as our next topic, but separate from here. All right, so harvesting stored energy. Glucose is the model. Catabolism of glucose is to produce ATP. Okay, so we have glucose. This is our cellular respiration equation. This is very important. So glucose, here it is. So it's the word equation, and then we're going to have our actual chemical formula equation. So glucose plus oxygen. So here we are, C6H12O6, that's glucose, plus oxygen, so 6 O2s because we're balancing this equation, yields, okay, so this should be over here, energy, which is ATP, water, and carbon dioxide. So ATP, our energy, 6 water, and 6 carbon dioxide, plus heat. Heat is always released as a byproduct. So this is our pretty soon you're going to be able to tell me where everything's used and where everything is produced. It's going to be really cool. We know this occurs in our mitochondria, and we're going to talk about that more. So combustion means making a lot of heat energy by burning fuels in one step. Okay, the fuel here is the carbohydrates. So combustion occurs with oxygen. Again, we talk about that. So glucose plus oxygen makes ATP. Okay, respiration, making ATP and some heat by burning fuels in small steps. So here we are, respiration. Oh, there's our enzymes and ATP. Okay. So how do we harvest energy from fuels? We digest large molecules into smaller ones by breaking bonds and moving electrons from one molecule to another. Okay. As electrons move, they carry energy with them, and that energy is stored in another bond and released as heat or harvested to make ATP. So we have an electron being moved from yellow to blue, and we're talking about oxidation and reduction. So here I'm going to remind you, loss of electrons is oxidation, gain of electrons is reduction. So we talk about this a lot. So they're called an oxidation reaction because it reflects the fact that in biological systems, oxygen, which always attracts electrons strongly, is the most common electron acceptor. We're going to talk about oxygen so much being an electron acceptor here. Okay, so it's oxidation, always accepting. Oxygen always is our electron acceptor. Oxidation and reduction reactions always occur together. Therefore, we kind of refer to them as redox reactions. And if you've ever had chemistry, or if you're in chemistry, you're going to learn about this a lot. So please remember, yellow has lost an electron, it's oxidized. Blue has gained an electron, it's reduced. This is really important throughout, okay? The reduced form of a molecule has a higher level of energy. So that electron has carried energy with it to this reduced molecule. Then the oxidized. So blue now has more energy than yellow. The ability to store energy in molecules by transferring electrons is called reducing power. It's a basic property of all living systems. Okay, so important information there. Oxidized, lost an electron has less energy. Reduction, gained an electron has more energy. All right, let's keep going. Oh, here we go. Sorry, I forgot these were here. This also goes ahead and refers to it's now charged. So this lost an electron is now got a positive charge. It gains an electron, now has a negative charge. Okay, so there we go. That's called a redox reaction. All right, so how do we move electrons in biology? Okay, so moving electrons in living systems, electrons cannot move alone in cells. They have to move as part of a hydrogen ion. All right, so energy is transferred from one molecule to another during these redox reactions. Okay, so when we move hydrogen, we move electrons. So here we go. Loses an electron, gains an electron, the same thing we saw before. So if we go down here, C6H12O6, which is glucose, has been oxidized fully. Okay. Each of the carbons has been cleaved off, and we're going to talk about this step by step. And all of the hydrogens have been stripped off and transferred to oxygen. So essentially, that's what's happening. Okay. So remember, oxygen is the most electronegative atom in all living systems, and this converts oxygen into H2O, water, as it's reduced. And now I'm giving you a general overview. We're going to go step by step, so I don't want anyone feeling overwhelmed. The reduced form of a molecule has a higher energy state than the oxidized form. Remember, we talked about that. Reduced has more energy than oxidized, okay? The ability of organisms to store energy in molecules by transferring electrons is referred to as reducing power. The reduced form of a molecule in biological systems is the molecule which has gained a hydrogen atom. So there's an example, NAD plus turns into NADH. And the NADH is the reduced version. And we will continue, I'll continue to point this out throughout, okay? Soon we're going to meet those electron carriers, NAD and FADH, when they're reduced, and we're going to talk about how they store energy and how they move electrons, okay? So we're talking about here, oxidation is occurring, and then here, reduction is occurring. It's actually very interesting, and it makes so much sense so we can put things together. All right. So coupling oxidation and reduction, and please don't feel, please take a breath and don't feel overwhelmed because I really am going to break this down for you. Redox reactions in respiration. They release energy as a breakdown of organic molecules. They're breaking carbon to carbon bonds, also stripping off electrons from carbon and C and H bonds by removing the H atoms. So when they remove these H atoms, they're removing, hold on. Okay, sorry about that. So when they strip off electrons from CH bonds, they're doing it by removing the H atoms. So this sugar molecule goes to CO2, okay, and the fuel has been oxidized. Electrons are attracted to more electronegative atoms. In biology, what's the most electronegative atom? It is oxygen. We've been talking about it. So oxygen turns into H2O, which is water. When you see H2O, that means oxygen has been reduced. We couple redox reactions and use the released energy to synthesize ATP. So oxygen is two oxygen atoms, and they're both looking for electrons. Okay, so yeah, that's how we do it. All right. Bear with me, guys. What's going on with my cursor? Here we go. Oxygen. All right, so oxidation and reduction. So we're taking a look, we're comparing the two. So why don't you just draw yourself a little T-chart here. So oxidation is adding an O, removing an H, loss of electrons, release of energy, and it's an exergonic process, okay? When we're talking about reduction, we're removing an O, we're adding an H, we're gaining electrons, we're storing energy, and it's an endergonic process. Okay, so go ahead and take a look at that. So oxidation, what's happening here? We're adding an O. Oh, look at that. We are removing an H. We see no H's here. We're losing electrons. We're releasing energy. Here, we take the oxygen, which is being reduced. We are removing an O. We're adding an H. We're gaining electrons. We're storing energy, and it's endergonic. Okay, more about that. So moving electrons in respiration, we have these things called electron carriers. These become really important for us. They move electrons by shuttling H atoms around, hydrogen atoms around. So NAD plus is oxidized version, and NADH is reduced. And FAD plus 2, and then FADH2 is the reduced version. So let's talk about NAD. So nicotinamide adenine dinucleotide, NAD, and its relative NADP, that P represents a phosphate, which you're going to meet in photosynthesis. They're two of the most important coenzymes in the cell, and they are coenzymes, okay? In cells, most oxidations are accomplished by the removal of H atoms, and both of these coenzymes play a role in this. Nicotinamide is also known as vitamin B3, just so you know. It's believed to cause improvement in energy production due to its role as a precursor of NAD, which is that important molecule in our energy metabolism. So NAD is our electron carrier. So when you increase nicotinamide concentration, you are actually increasing your ability to produce energy. Okay. Just so for the record, vitamin B3, you can find it in meat, peanuts, sunflower seeds. FAD is built from riboflavin. You guys have probably heard of that before, and it's actually referred to or also known as vitamin B2. You can find that in organ meats, almonds, mushrooms, whole grains, soybeans, leafy greens. Okay. It's a coenzyme critical for our metabolism as well. For us, we're going to learn they're electron carriers, essentially. And the entire process of cellular respiration and energy creation revolves around them. All right. So let's keep going. Oh, we have NADH. We're looking at the actual process. So we're carrying electrons through as a reduced molecule. So NADH will pick up some hydrogens and electrons and become, or NAD will pick up hydrogen and electrons and become NADH. Okay, look at that. It's got reducing power, like money in the bank. All right, so let's talk about the overview of cellular respiration. So there's 4 metabolic stages that we need to know. Okay, the first is glycolysis, and that's anaerobic respiration. Anaerobic means without oxygen. So anaerobic means no oxygen, so no oxygen is required. So here we are, glycolysis is when we have respiration without oxygen. It occurs in the cytosol, also known as the cytoplasm. We will talk about it as when glucose becomes pyruvate. So this is step number 1. Okay. The next 3 steps we refer to as aerobic respiration, which is respiration using oxygen and occurs in the mitochondria. We have pyruvate oxidation, okay, which is right here. We have our Krebs cycle, and then we have our electron transport chain. You guys are going to be pros at this very shortly, so bear with me. So here's our equation again. Glucose plus oxygen yields ATP, water, and carbon dioxide. And heat. Can never forget the heat. All right. So here we are again. What's the point? Again, to make ATP. So I talked about this in the last step. ATP synthase enzyme. I'm just bringing it up again to remind you this is very important in the last step of chemiosmosis. It's where we have hydrogen flowing through it. It changes. It essentially turns like a motor and then turns ADP into ATP for a short period of time until we try to lose that bad boyfriend. Okay, so how is the gradient formed? So ultimately, this is where we want to get to. How is this gradient formed? I want you to keep that in your mind. All right. Let's see where we are. So we're going to talk about stage 2 and 3. So this is the overview. Okay, the first 10 reactions, I'm never, ever, ever going to make you memorize what happens in each reaction. I do want you to know the different parts, the 4 different parts. Okay. So we have the first 10 reactions convert glucose into pyruvate, produces 4 ATP and 2 NADH, but it also uses 2 ATP. So essentially, we have a net of 2 ATP and 2 NADH. So 2 NADH being those electron carriers, these are the reduced form. Okay, this happens in glycolysis, where we have glucose out in the cytoplasm turning into pyruvate. All right. Glycolysis is only the start. So in glycolysis, we have a glucose turning into pyruvate. Sorry. This again occurs in the cytoplasm with no oxygen. The pyruvate isn't done here. It has more energy to yield. There's 3 more carbons that we need to strip off, or what we call oxidize. If oxygen is available, pyruvate is going to enter into the mitochondria. And then the enzymes of the Krebs cycle are going to complete the full oxidation of sugar into carbon dioxide. So because pyruvate has 3 carbons, all right, so pyruvate has 3 carbons, so there's still energy in those bonds. All right, so that takes us to our next step. Maybe. There we go. All right, so we're taking a look at this overview. We've talked about glycolysis, no oxygen present, glucose to pyruvate. If we have oxygen present, we will talk about the alternative when there's no oxygen present, but we are talking about oxidative phosphorylation, which is the other word for cellular respiration. So pyruvate is going to go right into the mitochondria and it's going to be oxidized and put into the Krebs cycle. So let us talk about that. So the mitochondria structure, it's got a double membrane and it's an energy harvesting organelle. It's got a smooth outer membrane and a highly folded inner membrane. If you take a look here, and we call those cristae. There's also an intermembrane space. So here's our between membranes. There's an intermembrane space. And then there's a matrix, which is in here. Okay. And that's filled with fluid. It's got DNA, it's got ribosomes, it's got enzymes. All right, so there's our structure of the membrane. And that's what it actually looks like under an electron microscope. Again, what cells do you think would have a lot of mitochondria? What do you guys think? Okay, dividing mitochondria. So they divide just like prokaryotic cells. Yep, bacteria. And what does this tell us about evolution? So we're not going to get into. So we had talked about briefly the endosymbiotic theory, where we thought that prokaryotic cells engulfed mitochondria and chloroplasts because they do have their own DNA and they do have their own mechanisms for reproducing. They reproduce independently of a cell. So that talks about endosymbiosis. So we have these membrane-bound proteins inside of them as well. Okay. And the advantage of a highly folded inner membrane is that we have more surface area for membrane-bound enzymes. So more surface area. Whenever you see a lot of folds like that, more surface area is better. And just to go back and answer our questions, the most mitochondria will be in our muscle cells and in our nerve cells. Okay. Form fits function. All right. So let's talk about the oxidation of pyruvate. Pyruvate enters the mitochondrial matrix. You should know where that is. We just talked about it. And becomes acetyl-CoA. It's a 3-step oxidation process. Here we release 2 CO2s, okay? And we reduce 2 NADs to 2 NADHs. That means we're moving electrons. And we produce 2 acetyl-CoAs, okay? CO2 is a fully oxidized. So here we are. We have the CO2 is fully oxidized. And because the body doesn't need it anymore, we get rid of it, okay? CH4 is a fully reduced carbon and it's full of hydrogen, so it's a good fuel. So we're going to talk a little bit more about that. We have acetyl-CoA entering the Krebs cycle now. Here's 3 carbons and now is 2 carbon. And we got rid of this carbon dioxide because this has been completely oxidized. It has no function for us anymore. So goodbye. And then we have an NADH, which is going to move an electron. So again, see, we keep showing this as money, but again, it's the energy currency where those NADHs get used elsewhere. Where does the CO2 go? We exhale it out. So when you guys breathe out, you are breathing out a byproduct of cellular respiration. Okay. All right. Pyruvate is oxidized into acetyl-CoA. So this is what we're taking a look at here. We have this pyruvate. It comes in. We release the CO2 because it's completely oxidized. It's already released all the energy that it can. So it's no longer viable for the cell. We don't like it. We don't want it. It's toxic for our cells. So CO2 is always being exited out through our body through sweat, through pee, through breathing. Okay. So we have produced an NADH and a CO2. All right. Pyruvate. This is our transport protein, our coenzyme. Excellent. So now we have acetyl-CoA. Acetyl-CoA is, yep, okay. So we're just taking a look of, because, matter can't be created or destroyed, it's just transformed. So we have 3 carbons going to a 2 carbon, but here's the excess carbon going bye-bye. Okay. Oxidation, reduction. So the carbon is oxidized, the NAD is reduced to NADH. There again is our piggy bank. I hope you guys appreciate these. All right. The Krebs cycle, it's also known as the citric acid cycle. It occurs in the mitochondrial matrix. It's an 8-step pathway that's catalyzed by specific enzymes. By the way, this is a dude, I think it's Hans Krebs. He, I think 1981, he lived to be 81, okay? And the Krebs cycle is named after him. Stepwise, it's the catabolism of 6C citrate molecule. Again, not something you need to know. This actually evolved later than glycolysis, so there's a lot of the enzymes of glycolysis are used, or glycolysis is used to study commonalities in organisms and how they evolved, because glycolysis occurs in bacteria. It's something called archaeobacteria, as well as eukaryotes, which is us. So they use that and study it. So it evolved later than glycolysis. I'm not going to spend a lot of time talking about this. All right. So we're going to count the carbons. Are you ready? We have a 2-carbon acetyl-CoA that goes into the Krebs cycle, and it's taken apart completely. We have 2 CO2 molecules out of here. Okay. It produced from that 2-carbon sugar, and now the glucose is completely oxidized. But again, where are all the ATPs? So we have the oxidation of sugar, completely oxidized. Get rid of the carbons. We don't need them anymore. Exhaled out of the body. This happens twice for each glucose molecule. Okay, so look at all these things that we produced. The Krebs cycle is extremely, never would I ever make you remember this. But what we have being made along the way, so we made a CO2 and pyruvate to acetyl-CoA, we made an NADH. Along the way, we make another CO2, another NADH, another NADH, another FADH, another NADH. So 1, 2, 3, 4, 5 electron carriers, all really important, but we still don't have the ATP. Okay. Why am I going back and forth? Reduction of the electron carrier. So again, all of these guys reduced, extremely important. All right, so we fully oxidized glucose, C6H12O6. We've used it up, right? We now have CO2 produced. And we ended up with 4 ATP here. Okay, that's still not enough. So every time the carbons are oxidized, an NAD plus is being reduced. So do you see how that happens? Oxidized carbon, reduced NAD. So redox reactions always coupled, always occurring together. Okay. All right. What's the point? Let's get to that point. So let's talk about our electron carriers. They also are hydrogen carriers. So the Krebs cycle produces large quantities of these electron carriers. NADH, FADH2. They all go to the next stage, which is our electron transport chain. So what is so important about these electron carriers is that they are going to provide the fuel for the proton pump. All right. So we're talking about the energy accounting of the Krebs cycle. We have made 4 NADHs and 1 FADH, and we have produced CO2 and 1 ATP. So we're still waiting for all of the ATPs, because so far we've made 3 net, I believe, for 1. So, 2, my bad. 2 net gain of ATP, and then 8 NADH and 2 FADH2. Do you have to remember those numbers? Not at all. But what I do want you to remember is that FADH and two FADH2 or FADH2 was made. All right. So if the yield's only 2 ATP, then how is the Krebs cycle and adaptation? So we're talking again about this value of NADH and FADH2. These are electron carriers and hydrogen carriers, okay? These are what move electrons and they move hydrogen ions to be used in the electron transport chain. So far, we've gone through glycolysis, pyruvate oxidation, the Krebs cycle, and now we're really going to get into the electron transport chain and chemiosmosis. So again, FADH and NADH, they're like our money in the bank. Yeah, okay. So how do we do this? Again, ATP synthase sets up a hydrogen gradient. We're going to allow hydrogen to flow through that ATP synthase. It powers the bonding of ADP and phosphate, and it creates ATP. Okay, so how are we going to get there? We haven't done it yet. All right. So let's go to stage 4, the electron transport chain, which might possibly be my favorite. All right, again, the overview. This is called substrate level phosphorylation. Here's our oxidative phosphorylation. Oxidative, remember what that word means. Okay, again, I know I keep bringing this up. I'm sorry, I didn't make this PowerPoint. So the ATP accounting so far, 2 from glycolysis, 2 from the Krebs cycle. Okay, 4 ATP is not nearly enough if you are burning 10 million ATPs in a muscle a second. All right. Yeah, there we go. We brought that up last time. So what the heck? There's got to be a better way, and there is. The electron transport chain is a series of proteins built into the inner mitochondrial membrane along the cristae. So you got to take a look at what the mitochondria looks again. I think I have a picture I'm going to pull up. Transport of electrons down this electron transport chain are linked to the pumping of hydrogen ions to create a hydrogen ion gradient. Okay. This yields a total of 36 ATPs from 1 glucose, and you only in the presence of oxygen will this occur. And let's talk about why. So here's what the electron transport chain looks like. This is the membrane, the inner membrane of the mitochondria. This is our electron transport chain. Okay. So what's going to happen? I'm getting ahead of myself. All right. So our mitochondria has this double membrane. The outer membrane here, and our inner membrane is where this electron transport chain occurs. It's highly folded for more space. It's got those enzymes and transport proteins. It also has an intermembrane space. So this is our intermembrane space here. Okay. It's fluid filled and it's between the membranes. Remember that, please. It's important because form is important to function always. So we're going to take a look at a cross section here. Here is what it looks like. This is the inner mitochondrial membrane. This is the electron transport chain, okay? This is the inner mitochondria. This is the intermembrane space between membranes. This is inside in the matrix. So here's our intermembrane space. This might be a really good thing for you to draw. So do take the time and do that. Here's our mitochondrial matrix. So these are the names of these proteins. I'm never going to make you memorize them, but I am going to tell them to you. So NADH dehydrogenase, can you work that name out? Cytochrome B C complex, and our cytochrome C oxidase complex. This little guy is called, I went too far. Boy. Let me go back. Okay, I can't go back. You guys can go back and pause it. So we're remembering the electron carriers from glycolysis, from the Krebs cycle, right? It's time to break open those piggy banks. So let's go ahead. Oh, we're back. Oof, thank God. What's going to happen is NADH is going to drop off its hydrogen and electrons. Okay. So we're going to get an electron and we're going to get a hydrogen. So NADH is going to be oxidized and become NAD. But what happens is their hydrogen is going to go up and their electron is going to travel through this electron transport chain. So as the electrons travel through this chain, what happens is it fuels the pumping of hydrogen ions out. Okay, look at that. Kind of cool, huh? All right, so we're getting hydrogen ions built up. Now remember that question that we asked, how do we get that hydrogen gradient, right? These are actually just protons, guys. So FADH now comes in, and FADH gets oxidized, and it drops off its electrons and its hydrogens. So we have even more electrons coming to fuel the hydrogen pumping, okay? So here we go. I want you to take a look at half of O2 is just O. This is our final electron acceptor. You are going to see that question a million times on your next quiz. Your final electron acceptor is oxygen. So the electrons now need to go somewhere, because if the electrons have nowhere to go, then this electron transport chain becomes backed up. And then we have no hydrogen ions, and then we have no ATP, and people die. So I want you to think about something for a moment. Why do we breathe? We breathe for this process right here alone, right? So if someone stops breathing, what is it in their cells that stops working? The electron transport chain. Why do they die? They have no ATP. Just, a little tidbit. So we have two hydrogens in here, and then we have a half of an oxygen. They get the electrons and they become H2O. Water is the product of the final electron acceptor of oxygen. Because remember, oxygen always wants its electrons. Okay? We're building the proton gradient. What powers the proton pumps? Okay. So, all right. We're almost there, guys, and you're doing a really good job. So I'm really proud of you. We're going to break this down big time in class. All right. So stripping hydrogen from electron carriers. The electron carriers pass electrons and hydrogens to the electron transport chain, like I just showed you. The H is cleaved off of the NADH and the FADH2. The electrons are then stripped from the hydrogen atoms, so they become protons, hydrogen protons. And those electrons are passed from one electron carrier to the next, like I was just showing you. The flowing electrons are what give us energy to do work. Transport proteins in the membrane pump the hydrogen ions, now protons, across the membrane to the space. Ta-da! Does this picture look familiar to you guys? All right. So here we go. We're looking at this happen like I just showed you guys. Now we have all these hydrogens out here. What's going to happen? They're going to flow in. This is going to turn, and then we're going to make some ATP. Moving electrons do all the work. Life literally depends on the movement of electrons, guys. That's why in integrated science, we're like, electrons, electrons. Why are electrons important? Electrons are important for everything, but there would be no life if there were no moving electrons. They are the best. So what pulls the electrons down? The flow of electron powers the pumping of them out. So what pulls them down? You guessed it, our highly electronegative oxygen is pulling those electrons down. It's always looking for them. It's like electron hungry. So like what pulls those electrons back down? It's the oxygen, because it's so electron negative. It always wants to go downhill to the easiest path, which is oxygen. So moral of the story, oxygen is greedy as ever for electrons. I hope you guys remember all these little stories I'm telling you. We call this oxidative phosphorylation. Remember, we learned what phosphorylation is. So phosphorylation is adding of a phosphate group, right? And then oxidative, oxygen. Yeah, okay, sorry. So electrons will flow downhill. And we're just taking a look here. They move from carrier to carrier downhill to the oxygen. Each carrier is more electronegative, and it's called controlled oxidation, okay? And it controlled release of energy. So as these electrons go, we have a controlled release of energy, and they're pretty low energy when they get here to form water. So we finally did it, guys. We did it. All right. We set up a hydrogen gradient. We allow the protons, which are these hydrogen ions, to flow through ATP synthase, which makes its shapes change, which synthesizes ATP from ADP, and we did it. This is referred to as chemiosmosis. All right, there it is, chemiosmosis. The diffusion of ions across a membrane, okay? More specifically, it relates to the generation of ATP by the movement of hydrogen ions, okay? across the membrane. So here we go. The actual chemiosmosis part is this buildup of this gradient and the moving through to create ATP. It links the electron transport chain to ATP synthesis. So there's the point. Peter Mitchell, the dude that figured it out, he's the one who proposed chemiosmotic hypothesis. Actually, you have a question about chemiosmosis on your next test. It's open-ended. Let's take a look here. Ready? 1. Electrons. This is our electron transport carrier. Electrons are harvested and carried to the transport system. Here, look where we're getting all our electrons from. NADH, NADH, FADH2, right? The point is for them all to go to the carrier. Electrons provide the energy to pump the protons across the membrane. Oxygen joins with protons to form water as our final electron acceptor, and the electrons are attracted to that water. The electrons are low energy here. We have the protons diffuse back down their concentration gradient, driving the synthesis of ATP. Voila. I feel like that's artwork in my own opinion. Okay. This is just another way to look at it. 2 ATP made here, 2 ATP made here, plus 30 around. So this number means around 36 ATP. This number is hotly debated. No one knows for sure. So roughly 40 ATPs are made for every 1 molecule of glucose you eat. Okay, here's our summary. Ready? Where did the glucose come from? You ate it. Where did the oxygen come from? You breathe it in. Where did the CO2 come from? It's the oxidized carbons cleaved off of the sugar in the Krebs cycle. Where did the CO2 go? We exhale it. Where does the water come from? Water comes from oxygen being the final electron acceptor combining with hydrogen and creating water. Where did the ATP come from? Well, mostly the electron transport chain. So what else is produced that's not listed? NAD is an end product, so the oxidized version of NADH. So NAD, FADH, and of course heat. Why do we breathe? You guys should know that. So I mentioned this before, and we're actually going to be doing a quick activity with this. What happens if there's no oxygen available? So what I was talking about before, when people suffocate or people drown, that means they have no more electron acceptor. So what's going to happen? The electron transport chain backs up. Nothing to pull the electrons down the chain. NADH and FADH can't unload their H. And ATP production ceases, and it takes minutes for the body to die. Okay? The cells can run completely out of energy, and everyone dies. Very sad story. Cyanide is something that does that, by the way. Cyanide poisoning, it takes out oxygen and it backs up the ETC. We're going to do some work with that. Now you know the point. We are not going to talk about this right now. Okay, we're going to stop. All right, guys, great job.